Hello and welcome everybody to this research seminar from January 2022. I'm very pleased today that we have Professor Andrew Wilkie here to talk about craniosynostosis and Professor Stefan Marcianak and Fiona Copeland here to talk about pneumothorax management and particularly around the PPI side of that. So how we're making sure that participants and their views and their input can help to make the research that you guys are doing even richer. So there's a few slides that we're just going to run through first of all, and then we'll get started on the talks. I'd like to remind everybody that this is a recorded event and it will be available on Genomics England's YouTube channel in due course. So if you have any comments to make in the chat, that's all very welcome. And if you've got any questions that you'd like to put to our speakers, please can you put them in the Q&A section and we'll make sure that we address them if we can in the time available. The format today, oh sorry, yes, I may want to introduce myself. Sorry, I forgot that bit. Yes, I'm Jillian Hastings-Ward and I'm the participant panel chair at Genomics England. So I am a participant in the 100,000 Genomes Project because my son, who's severely disabled, was registered for it back in 2015. I represent the people whose data you are playing with and looking at and investigating in the course of all of your work in the research environment. So we're very grateful to you for all the work that you do, and we're keen to hear even more about it and the outputs of that as we move forward. So the data that you're looking at came from the 100,000 Genomes Project initially, and this has always been free to access for clinicians across the world. And if you wish to join in with the work of the GSIPs, you need to make sure that your institution is registered with Genomics England as an accredited partner. Researchers in the industry can apply to join in via the Discovery Forum. If you have any questions about how that can be made to happen, do get in touch. The address there is on your screen. Gzip Help at Gzip-help at genomicsengland.co.uk. We'd like to make sure that these seminars focus on the stories from you about how you've been working with participants and their representatives in the research that you've been undertaking so far. And so if you have any tales that you wish to tell and practices that you'd wish to share, or indeed challenges that it would be helpful to surface with the team, they'd love to hear from you with the potential of speaking at a seminar in the future. These events are very much designed around the needs of your community, and so we're keen to make sure that we talk about issues that matter to you. Our speakers today, Andrew Wilkie, Stefan Marcianak and Fiona Copeland, will be taking us through their specialist work that they've been doing on the data which has been collected so far. And I'm really looking forward to hearing from all of them because their insights will really help us understand more about what's been going on and what's going to happen next. Next month, we have another research seminar at the same time on the 22nd of February. The topics covered that day will be the use of polygenic risk score prediction, or the use of whole genome sequencing data in predicting polygenic risk, and also colorectal cancer. So again, two very different topics, but I'm sure they've both got a lot of interesting insight that they've been able to make from the data so far, and do check in with that one. There's a list of resources here on your screen. Lots of these things are available through the Genomics England website and the research community newsletter, which came out recently. Hopefully many of you will have received a copy of that. Again, contact the GSIP team if you'd like to be added to their mailing list, because it's a very rich resource full of really useful information and project snippets about what's been going on recently and what Genomics England are doing to try and make it easier for you guys to use this information to your advantage. There's two other big events we want to tell you about today. The first one is a research summit, which is coming up on the 4th of May. This is the successor to an event which was very successfully held pre-COVID back in November 2019. And I'm not sure, perhaps many of you on this webinar today were there on the day, where we heard lots in person from the different researchers around the world who are using this data. The next one is going to be on May the 4th. Hopefully it will even be in person. Imagine that. And if you'd like to get involved, please do contact the events team at Genomics England, especially if you have an abstract that you wish to share either as a presentation or as a poster. I believe the deadline for initial submissions is the 11th of February. So do get your thoughts together by then if you can. The other event to tell you about is the Sight Loss and Your Genome seminar coming up this Thursday, prepared in collaboration between the researchers looking at the sight loss data and the participants in the participant panel and others who have been keen to try and broadcast more news about what's been happening within this Gzip. So that one is also freely available to attend. And again, if you'd like to contact the Gzip team, they can help you get into it. So I think we may as well move forward. We were hoping to hear also from Dr. Richard Scott, who's the Chief Medical Officer at Genomics England, just to say another few words before we get into the main speeches, but I haven't heard from him. So I'm here today. Oh, you are here. I'm so sorry, Richard. Didn't see you arrive. So would you like to just say a few words before we hear from Andrew Wilkie?
Yes, thank you. Thanks to you, Gillian, for hosting, and to everyone for joining, as well as our three speakers today. We're really grateful for your time and really looking forward to listening to it. Thank you, Andrew, particularly for bringing your talk today. You'll hear from Andrew. This is while it's been powered by Andrew's team and his work in terms of making these discoveries. It's been something that we at Genomics England have been really keen and happy to be part of, and we've learned lots as we've gone along, and you'll hear that. And I think thank you both for the diagnoses and findings that have been found through this work for individual families. At my hospital, that's been impactful for families that we've seen, but also it's been really important in terms of helping us to plan changes in terms of our pipelines and improvements as we've been through the program, but also for the genomic medicine service. In fact, we've got a couple of people who have joined, will be in the chat today, if that's useful, to sort of answer questions about some of the learnings in terms of impact on our pipelines. It's also been really helpful thinking about how we make sure that it's not just in areas like craniosynostosis, which is Andrew's area of special impact, where families benefit from this sort of fine tooth comb look. And the team internally are looking at individual families with some of these exact learnings. And again, Susie, who's on the line, is doing some of that work at the moment, and as well as thinking about how we can do that at larger scale and coordinate with other Gzip teams to make sure that this is sort of something that doesn't require just the brilliance of Andrew's team, but is something that we can make sure everyone benefits from who can. Just wanted to sort of say thank you in advance for that, and all thanks to the other speakers as well.
Thank you, Rich. Great. So I'd like to introduce Professor Andrew Wilkie. I'm sure many of you have encountered him before, but for those who haven't, he's an honorary consultant in clinical genetics at Oxford University Hospitals and the Nuffield Professor of Pathology at the University of Oxford. He works closely with plastic surgeons, and his clinical research aims to identify the molecular genetic basis of craniofacial malformations, particularly craniosynostosis, which is the premature fusion of baby skulls. Over the years, his group have identified many new disease genes, and his team was the first to demonstrate the diagnostic value of exome and genome sequencing in relation to this condition. Extending this work to the 100,000 Genomes Project data, they uncovered several reasons why clinical molecular diagnoses had been systematically missed by filtering pipelines. And his current work explores the use of optical mapping and nanopore sequencing to find further copy number and structural variants. So Andrew, welcome to the seminar. You have 25 minutes, and then we'll take some questions. And if anyone has questions as we go along, please put them in the Q&A box rather than the chat box, so that we can find them quickly. Thank you very much.
Okay, thank you very much for that introduction, Gillian, very kind. And can you see my screen, first of all? That's working okay. So as you've already heard, I'm a clinical geneticist, but I specialize in this specific condition, craniosynostosis. And I work very closely with a multidisciplinary team at the Oxford Craniofacial Unit. So I'm going to talk about my experience of 100,000 Genomes Project data applied to this particular condition. So first of all, what is craniosynostosis? Let me just get my... So it's the premature fusion of one or more cranial sutures of the skull, and it's got a prevalence of about 1 in 2,000. So between 300 and 400 babies are born with this every year. The sutures enable the narrow gaps between the bones that enable expansion of the skull during childhood, so the brain can grow, obviously. And if one of these sutures fuses prematurely, then the skull overgrows at the other sutures, and this gives rise to characteristic distortion of the skull shape, as summarized here. And we investigate this primarily using 3D CT scanning. And so here are scans from a couple of children, one of whom has just a single suture fused, as shown here, compared with the normal suture on the other side. And then this is a child with a scan shortly after birth, and you can appreciate that this child has an extremely severe form of craniosynostosis called cloverleaf skull, where all the sutures are fused before birth. So it's a very variable condition. So there were a number of reasons, I think, why craniosynostosis provided a good model to look at the use of 100,000 Genomes Project data. Surgery is centered in England at just 4 units. And the clinical geneticists at those 4 units know each other very well. And we have a culture of sharing clinical data with each other. All those geneticists were already participating in a genetic study that I set up in 2009, genetic basis of craniofacial malformations. And that meant, as it turned out, that around about 40% of the patients enrolled into 100,000 Genomes Project were already in our study. There's also a comprehensive NHS genetic testing service for the 7 genes most commonly mutated in craniosynostosis. And there were strict clinical criteria to recruit only those patients who are most likely to have a missed genetic diagnosis to 100,000 Genomes Project. So all that meant that the patients who were enrolled into the 100,000 Genomes Project with craniosynostosis, there were no kind of easy wins with them. And that's what whole genome sequencing should be doing, to look at the difficult diagnoses. And then in addition to that, in my laboratory at the Weatherall Institute of Molecular Medicine, I had a dedicated team of researchers to scrutinize these data in detail. So if we look at the organization of the 100,000 Genomes Project, so Genomics England, responsible for the sequencing, variant calling and prioritization, would then feed that data into the NHS clinical service, summarized here. But in addition to that, a research environment was set up to contain these data with the primary aim of identifying new causative genes and genetic diseases. And I'll present a little bit about that at the end. But what it also could be used for, as it turned out, was what's called diagnostic discovery. And so this is where researchers are assisting in making clinical molecular diagnoses. So how does this work? Well, you've already heard Jillian mention these Gzips, of which there are over 40, one of which is musculoskeletal, which craniosynostosis sits in. And so the idea would be that researchers could access the data within the research environment. And then if they detected a variant that they thought might be causative in a particular family, they could enter what was called a researcher identified potential diagnosis, or RIPD, and this could then be fed back into the NHS system. And so my group looked specifically at the data for the craniosynostosis patients in the 100,000 Genomes Project. So these RIPDs, this was the mechanism set up, and this was the form that you had to fill in, and we filled in several dozens of these forms, and nothing very much seemed to happen to them. There didn't seem to be diagnoses coming out the other side from the labs. And so I decided, in consultation with Richard, who you've already heard from, to look into this in some more detail. And this is basically a project that was facilitated by us all being locked down from March 2020, because this is something that could very well be done remotely. So I'm always going to think of this particular piece of work as the lockdown project. And the people who actually did the hard work from the GEL's point of view, I particularly want to acknowledge two people, Zareen Haider, who pulled the original data set together, and Helen Britton, who followed up a lot of the findings. And then from our lab in the WIM, Edu Kalpena was really the first person to say, gosh, there seem to be a lot of missing diagnoses. And then two DPhil students, Becky Toos and Yang Pei, who respectively worked on looking at small variants and copy number and structural variants. So this work is actually published as a collaboration with Genomics England. It came out at the end of last year, so you can look up all the details in there. Obviously, I'm just going to give quite a quick and superficial presentation. And in addition, Becky wrote up a piece about this work in Headline News, which is the magazine for Headlines, which is the patient support group for individuals with craniosynostosis. So to cut a long story short, there were 114 families with craniosynostosis in the main program of the 100,000 Genomes Project. And of those, the GEL GMC diagnostic system found likely pathogenic or pathogenic diagnoses in 16 out of that 114. So our lab, together with other methods of identification, which used our contact with the clinical geneticists, identified a further 18 diagnoses in this cohort. And I should emphasize that by and large, these are clinical molecular diagnoses. They're with one or two exceptions, not research findings. So that the implication of that, I thought, was really quite serious, which was that over half of the diagnoses that were supposed to be found in this group of patients in the 100,000 Genomes Project were actually being missed by the system that had been set up to find them. So obviously it's pertinent to ask why those diagnoses were being missed. And so this pie chart, which is in that paper I mentioned, summarizes those reasons. And so each of those sort of 18 missed diagnoses is represented by a gene name or chromosome position. So that's shown here. And this is looking at lots of different approaches to diagnosis and which ones of those should really have picked up a particular gene. And so it's a complex interlocking set of circles in a Venn diagram. But so I'd just like to emphasize 4 of the major reasons. So the single largest reason actually was not systematically looking at and scrutinizing de novo mutations, because a lot of those originally were put into what was called tier 3, and the labs weren't mandated to look at those. The next was that there was no system for systematically updating panels following the original examination of the data for a particular patient in the light of further discoveries as the 100,000 Genomes Project went on. The third was that even in the absolute top tier of genes, the tier 1s, we found that quite a number of diagnoses were being missed because variants were being filtered out from those so-called green genes. And then finally, that although we do genome sequencing in large part because it can look at bits of the genome that can't be seen if you just look at the genes in an exome. In fact, the way that 100,000 Genomes Project was looking at the genome missed a lot of relevant structural and copy number variations. So just to kind of go through that again, now I've sort of summarized those and say, well, what's happened since then? Well, it's certainly been recognized now, because now we have the new NHS Genomic Medicine Service, that it's important to look at de novo mutations. So that one I'd say has been sorted out. And I'm told that in the new NHS Genomic Medicine Service, variant calling, particularly for a kind of variant called an indel, where you have a few nucleotides deleted or inserted, is better. I think there's still a question of how data are going to be returned to in an iterative fashion. And there's certainly still a question, which I'll mention a bit more at the end, about how these copy number variants and structural variants should be looked at. So I'll just give you a couple of specific examples from this grouping, just to sort of show you some of the issues that were involved. This is a list of clinical features in one of these patients, and actually, though I say it myself, I was the key person in this particular case, because and so this was during lockdown. I was looking at these features, and it mentions craniosynostosis, and it mentions postaxial polydactyly and syndactyly. So if you see that combination, that's basically pretty diagnostic of one condition, Carpenter syndrome. And then you can tell where the patients come from the number. And this number starting 117 were an Oxford number. So I thought, so here's an Oxford patient with Carpenter syndrome. And although I didn't know they were in the 100,000 Genomes Project, I remembered that one of the consultants in Oxford, Usha Keeney, had asked my opinion about a patient with Carpenter syndrome in whom they couldn't make a molecular diagnosis. So I thought, oh, I really should be able to find out what's going on here. So I scrutinized the Exomiser data, and at the 96th position in the ranking, I noticed there was this missense variant in a gene called MEGF8. And this immediately sort of excited my interest, because our group was the one to describe MEGF8 mutations as a cause of Carpenter syndrome. And actually the very first mutation that we ever identified was this one that was listed in Exomiser. So we knew that this must be a pathogenic variant, but this is a recessive condition, and this was a heterozygous variant. And so only the other variant didn't come up in the data anyway. So I got in touch with Yang and Becky and said, can you have a look at this gene in this patient? Because there must be a second variant to cause the Carpenter syndrome. And they first came back to me and said, well, we can't find anything. So I said, we'll go and have another look because there's something there. And so at that stage, Becky actually went back and looked at the original, what's called the VCF file, the list of variants. And there indeed was another variant in MEGF8. And it's a 3 nucleotide deletion shown here. And when you looked at the what's called the IGV plot, so these are the individual reads from the Illumina sequencing, it was very obviously there and very obviously correct, and it had been filtered out in a later filtering process. And so this discovering that these kinds of what, as I said, called indels had been filtered out, this prompted Becky to search for other filtered out indels in our cohort of 114. And she found 2 more. So just in this small cohort, 2.6% of diagnoses had been missed because of this filtering process. And so if you just extrapolate that to the whole of the rare disease program, potentially nearly 1,000 diagnoses could have been missed through this process. So here's another example. And this again was a family that was known to us because this is from Louise Wilson, the consultant geneticist at Great Ormond Street. And this family was already enrolled into our genetics of craniofacial malformation study. So Edu took a look at the structural and copy number variant data at regions that were known disease genes in craniosynostosis. And one of those genes is called TWIST1, and it's on the short arm of chromosome 7. And he noticed that there were a whole number of calls in the 2 data sets that are available. One's called MANTA, and the other's called CANVAS. And so you can see there's 1 CANVAS call and 4 MANTA calls in this region of chromosome 7. But the problem is that none of these calls actually intersected the TWIST1 gene itself. And so these calls were completely ignored by the existing system for variant calling, which actually only uses CANVAS anyway. It doesn't use the MANTA calls at all, even though, as you can see, MANTA has got far more calls than the CANVAS has. So then Edu used these calls and looking at the IGV plots to try and work out what was going on in this family. And you can see there these MANTA calls called INVs, that's inversion. And so he worked out that there was a huge inversion of this region, 13 megabases, but TWIST1 wasn't actually disrupted by it. So it wasn't called by the system. And so, I mean, you could say, well, this is just speculative, but because we had samples from this family through our study, we were able, or Edu was able directly to use what's called PCR to confirm that his predictions of the structure of this rearrangement were correct. And it was subsequently Louise organized for a standard karyotype to be done, which confirmed that there was this massive inversion present. And even now, these 2 examples, I'm told, the Carpenter syndrome and this twist rearrangement would not be detected by the existing diagnostic methods in the NHS Genomic Medicine Service. So that's an interesting point for discussion. So I want to just move on to the other side of this, which is actually trying to generate new research findings. And so talk very, very briefly about the 78 cases with no causative variant identified. And as we mentioned in our paper, there's another process called a contact clinician request form. So this is where you don't think you've got a clinical diagnosis, but you've got some sort of interesting thing that you would like to follow up further. And so I'd say this process worked rather better than the RIPDs. And so here's a case where Edu found something interesting. And this is what's called an analysis of homozygosity. So this is looking at regions in consanguineous individuals, and here the parents were half first cousins, where essentially the same block of chromosome has been inherited from both parents. And so in a particular child, Edu noted that there were these blocks of consanguinity, including a variant in a gene called SPRI1, which encodes Sprouty1. And the reason he was interested in this is that this Sprouty1 is a negative regulator of signaling through fibroblast growth factor receptors, which are frequently mutated in craniosynostosis. And a knockout of Sprouty1 in the mouse shows kidney and urinary tract anomalies and craniofacial size reduction. So through this contact clinician process, we were able to make contact with Emma Wakeling, who's a consultant clinical geneticist also at Great Ormond Street. And we got consent for samples, to collect samples for our craniofacial malformation study. Becky then confirmed that indeed in the child, there was an early nonsense mutation in both alleles of the Sprouty1 gene. So this would stop the protein being made at this position. So here's a picture of the child. Obviously, consent's been given for this, illustrating a number of unusual dysmorphic features and listing the other features present in this child, an important one of which was bilateral cochlear dysplasia, leading to hearing loss. And then subsequently in cells, Becky demonstrated that Sprouty1 protein is completely missing in this child. So this child is the first naturally occurring knockout of Sprouty1 in humans, as far as we know. And so that's obviously important information for the family, and also moves biological knowledge a little bit further on. Now here's the other example that I want to show. And so this is now moving into the region of copy number and structural variants. Another craniosynostosis trio, this one from Guy's. And what Edu noticed, so this is a plot of read depth in the mother, the proband, and the father, was that there were two regions shown here where the read depth increases in the proband compared with either parent. So it looks like these are two de novo duplications, and they're on the same chromosome, chromosome 13. Again, through this process, we were able to make contact with Francesca Fulzano at Guy's. Yang obtained samples and then showed that you could verify the breaks in this patient. But in fact, with these two duplications, they're two alternative structures for how they can be orientated. One like that, and this is well away from this gene of interest, fibroblast growth factor 9. But the other one is, if I can get this to work, like this, and this time it's much closer to fibroblast growth factor 9. And the significance of that would be that this would bring it within what's called a topologically associated domain. And it's much more likely that FGF9 function is going to be affected by this structure than by this structure. So it then becomes imperative to work out which structure is present, but this can't be done using short read sequencing. So we're using two methods for long read technologies to try and deconvolute cases like this. One is called BioNano, which is an optical genome mapping method, and we have an instrument in the WIM. And the other is called Nanopore sequencing, and we have a collaboration with the 100,000 Genomes Project, which is supervised by Greg Elgar. And so what we're doing is we're comparing these two methods to see if we can find other causative structural variation. And I'll just show you one example. This is very early data. And this shows a deletion that was detected in the 100,000 Genomes Project. It's a de novo deletion in this child. We don't actually think it's pathogenic, but we were interested to see what this looked like on the optical mapping and the nanopore sequencing. So here it is on the optical mapping. This basically provides like a very detailed chromosome banding or barcoding of the two chromosomes. And what it's worked out is that there's a piece missing from one of these two chromosomes, which corresponds exactly with the position of the deletion that was shown on the sequencing. And then here it is on the nanopore sequencing. This is the original Illumina short read 100,000 genomes data, and it is detectable, but you can see the read depth is very wobbly, whereas in the nanopore sequencing, it's absolutely crystal clear. So this deletion is certainly well detected by both these long read methods. And then here's just an example of something that the Illumina sequencing in the 100,000 Genomes Project was completely unable to detect at all in the sequence, and yet it's perfectly obvious a little duplications there in the nanopore sequencing. So we're aiming to recruit 15 to 20 unsolved 100,000 Genome Project families to ask how frequently these previously undetected rearrangements are as a cause of disease. So because you need this high molecular weight DNA, it does require fresh blood samples. And 9 families have been recruited to date. And this, I think, raises an interesting question because the going forward, the NHS labs collecting DNA, it's not clear that actually the DNA, the methods of collection are suitable for these type of technologies, which seem to me to be the future for disentangling these complex structural rearrangements. So I'll finish there. I'd like to acknowledge my funders and just leave you with a couple of thoughts. First of all, I hope this talk and the other you're going to hear demonstrate the critical importance of researcher access to maximize diagnoses for patients. And then, despite this, in our new NHS Genomic Medicine Service, which has been running for well over a year now, to my knowledge, there is no researcher access to those data, even though a large proportion of the families are signing up to it. And I think that's a shame. So I'll finish there and thank you for listening.
Thank you very much, Andrew. That was fascinating. Even as a non-technical specialist, I could understand the main points that you're making there, and thank you for sharing the details. We have a few questions in the Q&A. Some of them relate to points that you raised earlier in your talk, and some of them are more recent, but we'll start at the top. David Curtis asks, as de novo variants are important, will there be more emphasis on sequencing trios rather than individual subjects moving forward?
Yeah, so certainly that is the preferred design in and the recommendation in the new NHS Genomic Medicine Service. And I mean, I think you know, during the 100,000 Genomes Project, it was already recognized. This goes back to 2010 and Han Brunner's original work on exome sequencing, you know, the advantage of sequencing trios to find de novo mutations. So I wouldn't say the problem was so much that trios weren't being sequenced. It was that the trios were being sequenced, but then the de novo mutations were not being systematically looked at in the diagnostic pipeline. That was the main issue. And as I say, I do believe that that particular issue is being sorted out in the new NHS service, but actually what is still unclear is the extent to which these missed de novo mutations are still in the 100,000 Genomes Project data. And one of the things that's happening now is that geneticists and pediatricians are re-enrolling 100,000 Genomes Project negative patients into this new service to have essentially the same sequencing done, when actually we know that the analysis of the original data hasn't been adequate up to now.
Thank you. Yeah, that's something which we need to do some further work on, I think. Yeah, because the people who consented to give their data for the original 100K project expected that that data and those samples would be useful in perpetuity rather than needing to be refreshed in due course. Thank you. Next question. Well, quite a few people were interested to know why the indels had been filtered out initially. Somebody wondered, Jing Yu wondered whether that was because they'd lacked proper annotation, or if there are other things that you would like to say about the reasons why these gaps initially had been there.
Okay, so I mean, as I understand it, it's simply the way, in a sense, the knobs were tweaked in terms of what were the, you always, when you're doing filtering, you're always having to balance letting sort of through true calls and not letting through too many false calls, which then overburden the system with incorrect variants. So it's always a balance in these things. And so the problem was that too many of these true calls were being filtered out. I said even the one I demonstrated would still be filtered out because it's in a short section of repeat. So, I mean, that question I know has been looked at in applying the filtering to kind of standard genomes. And it did turn out that nearly half of all indels were being filtered out. So, I mean, there's recognition that this is a problem. And so, I mean, I know that a system is in process to try and address that. And I would, as I say, I predict that probably many hundreds of additional diagnoses will be made just by finding these indels systematically.
Thank you. Next, we've got two questions left and then we must conclude. The next question is from Sarah Pickett, and she says, a lot of your diagnoses depend on gaining access to DNA samples from the patients and their families. How easy was it for you to make the connections to the clinicians who submitted the samples to Genomics England and subsequently to gain access to the samples?
Yes, well, I think that's a very good question and well taken. And so because, as I described, we have this very sort of centralized system for craniofacial surgery. We were very lucky, really, because I was in many cases able to bypass the whole, what was actually, to be honest, a rather kind of creaking system, and sort of talk to the clinicians directly, because I knew who they were, and they could tell us who their 100,000 Genomes Project patients were. And because we had this already ethically approved study, Genetics of Craniofacial Malformations, we did have a means both to link up numbers, because of course everything is quite correctly anonymous in 100,000 Genomes Project, actually work out who those people were, and through a sort of somewhat cumbersome process, we were able to realize that actually many of these numbers were people in our own study. And then for those cases not recruited at the craniofacial units, as I say, there has been this contact clinician process, which in my experience works fairly well, although sometimes a problem obviously with a sort of legacy data set like this is that the clinician may have retired, for example, and then it can get to a bit of a dead end.
Yeah, thanks very much. There's some interesting comments coming through in the chat as well from people associated with the Genomics England team who are working on identifying new diagnoses. including de novo variants and indels that have been filtered out. And they're reporting that hundreds of variants are now flowing back through the diagnostic discovery pathway. So thank you for that.
Yeah, well, that's good to hear.
Yeah. Annalisa Taylor-Tavares from the team at Genomics England specifies that they've contacted clinicians with 580 clinical collaboration requests in 2021 and returned 422 potential diagnoses to genomic lab hubs for reporting since July 2021. So it sounds as though things are definitely moving in the right direction there. Thank you. Now, the final question on this section is from Ted Rees. He says, how much do you think these problems are common across all GSIP areas? And does this lead us to think that the sequencing prescribed in the NHS test directory needs further refinement? That's a very big question, Ted. We may not have time to cover it all. But Andrew, what's your thoughts on that briefly?
Well, many of the issues that we identified are systematic. Craniosynostosis is just an example condition. So provided those other disease areas actually have genetic causation, of course, some of the disease areas in 100,000 Genomes Project were a bit more speculative from that point of view. But if they have genetic causation, then something like missed indels or missed structural and copy number variants, those are likely to apply pretty much across the board. So I would say this has relevance to all disease areas, and those numbers that you're hearing of sort of additional diagnoses flowing back, I think probably supports that. Right,
thank you very much. And thank you all round, Andrew, for your very interesting presentation and really helpful answers. I hope you can stay with us for the second half.
Of course, yeah.
Thank you very much. Now, moving on, I'm delighted also to welcome Professor Stefan Marcianak and Fiona Copeland, BEM, who are here to talk about combining clinical, radiological and genetic approaches to pneumothorax management. Stefan is the Professor of Respiratory Science at the University of Cambridge, where his lab studies the role of abnormal protein folding in lung disease. He's also an honorary consultant respiratory physician at Addenbrooke's and Royal Papworth Hospitals with a clinical focus on pleural medicine. He's the subdomain lead for familial pneumothorax within the respiratory GSIP, and he's director of the NHS Familial Pneumothorax Rare Disease Collaborative Network. Fiona is the patient representative for the respiratory GSIP. She's a very well-known patient advocate for rare diseases, and she was awarded the British Empire Medal for chairing the PCD support group and raising awareness of PCD in 2021 New Year's Honours. She promotes, chairs and directs a large variety of national committees, groups and advisory boards working to promote knowledge and development of genetic disorders, blindness, cancer support and health research, and she's co-written numerous academic papers on PCD. So we're very much looking forward to this double act, hearing about how researchers and participant representatives are working together. Over to you.
Thanks, Jillian. Am I sharing the right slide?
I can see your title slide, thank you.
Fantastic. Well, I've changed the title slightly from the rather dry one about pneumothorax to calling it Punctures and Predicting the Future, because we're interested in punctured lungs, pneumothorax, but we're less interested in the effect on the lung, but more interested in using it as an opportunity to predict the problems the patient will have later in life, which are always more important than the pneumothorax. What we're going to do today, this is really going to be a focus rather than on the science behind pneumothorax, but more about the role of patient advocacy and also patient involvement in the development of this study. So I'm going to be giving an introduction first half. I'm going to talk for about 12 minutes on pneumothorax and the familial GSIP, but more importantly, Fiona is then going to talk about from the patient perspective. So just to set the scene, the most important person in all of this is the patient. And this is a sort of patient I would see in my pneumothorax service. This is an actual patient who's given the permission, who gave her permission for her case to be presented for teaching purposes. So she was a 68-year-old lady, which makes her slightly old for a patient in the pneumothorax service. As you'll see, they're usually younger. She presented to the emergency department in Cambridge with breathlessness and chest pain. And she immediately knew she had a pneumothorax because she'd been having them for 20 years. She'd had them since her early 50s, and in fact required surgery by the age of 53 to try and prevent them happening. But you'll see from this Richard's radiograph the surgery was only partially successful. She's got slight pleurisy. The lung is tethered at the apex, but she still has a large pneumothorax at the left base. Why had the cause of these pneumothoraces not been investigated and determined? Well, I can say that she wasn't seen previously at Cambridge. She wasn't actually a patient in the UK. She had returned from working for most of her life overseas, where the pneumothoraces had happened. The most important thing, apart from putting the chest drain in to reinflate the lung, was to ask her medical history. And remarkably, she tells us this hadn't been investigated previously. Her brother had started having pneumothoraces in his 40s, so this makes this familial pneumothorax. Her mother had started having pneumothoraces in her 60s. Again, this is familial pneumothorax, and this is not a family of heavy smokers, so this is not secondary pneumothorax. And her maternal uncle had also started having pneumothoraces later in life. So this is really obvious familial pneumothorax, but up until this point, despite having had pneumothoraces for over almost 20 years, nobody had attempted to investigate. As you'll see, in some cases, it's very easy to make the diagnosis if at least people start thinking. So this woman had a CT scan, and I've reconstructed this coronally, and this really makes the diagnosis to somebody interested in pneumothorax genetics. She has these cystic lesions in the base of the lung, and medially, they're sort of raggedy, non-symmetrical cysts in a basal distribution medially, which really rings the alarm bells for a condition called Birt-Hogdebay syndrome. And Birt-Hogdebay syndrome, when I'm talking to respiratory doctors, I always say it's the most common genetic disorder they've never heard of, because it is the most common genetic cause of pneumothoraces, and most doctors don't seem to realize this. She had other features of the syndrome when examined. Some of them are quite subtle. So these little white papules on her face are somewhat subtle, and they've been dismissed by her for most of her life as whiteheads. But these are obviously a small benign tumor of the hair follicle called a fibrofolliculoma, which is seen in this condition. Sometimes they're quite obvious and large and almost disfiguring, but usually they're quite subtle. But again, we don't really mind that she's got these small skin lesions, and we actually don't mind so much about the skin, the pneumothoraces, because we can fix them. What we mind about is that 30% of patients with Birt-Hogdebay syndrome go on to develop kidney cancer. And it tends to have, if the pneumothoraces start in the 40s, these tumors tend to start in the late 50s or 60s. So an early appropriate diagnosis gives the opportunity to enroll these patients in annual renal imaging, during which we can identify these tumors when they're very small. And when they're very small, when they're less than, say, 3 centimeters, they're relatively easy to cure by our urology colleagues doing partial nephrectomy. This patient was completely asymptomatic when she presented to us. Nevertheless, we started imaging, and she'd already developed a large tumor at the right apex as a renal cell carcinoma at the apex of her right lung. This could have been diagnosed when it was much smaller and much easier to treat, had the opportunity to make the diagnosis been made in time. So this is a breakdown of sort of the last 500 patients going through the pneumothorax service, and you see it's heavily skewed towards youngsters, sort of people less than 40. And these are typically tall, thin young men who have a tendency to develop spontaneous pneumothoraces. If we look at those 10% who have a family history, they're distributed more evenly through the age groups, although we do tend to see lots of youngsters. If we look at the sort of clinical diagnoses that we make that are then confirmed by genetic testing, this is the sort of the summary we see. Half of the patients will have this Birt-Hogdebay syndrome, the one that I've mentioned in our previous patient, but then a number of other syndromes, usually due to defects in the connective tissue, Marfan syndrome being the most famous and the one that every medical student suggests when they come to my clinic. But there are others, such as vascular Ehlers-Danlos loss, Loeys-Dietz, and increasingly rare ones. So Rachel Scott, a medical student actually working with us some years ago, codified the approach to systematically analysing patients with pneumothoraces. And the first thing is to ask if they have a familial pneumothorax, and that's 10% of the patients. And then we can divide them into 3 large groups of diagnoses or lack of diagnoses. There are those patients who have a defect in a component of mTOR signaling. You'll recall mTOR is a kinase that regulates protein synthesis and growth, And if it's deregulated and allowed to be too active, it tends to lead to at least benign tissue tumors, but occasionally malignancy. And Birt-Hogg-Debye is caused by a loss of function mutation in folliculin. One of the functions of folliculin is to regulate mTOR signaling. LAM stands for lymphangioliomyomatosis, which is a cystic lung disease which can be sporadic due to somatic mutations in TSC2 usually, but sometimes TSC1. But it's seen in about half of patients with tuberous sclerosis, which is due to germline mutations in TSC1 or TSC2. So this is one group. The other large group are defects of connective tissue. If you have humans or mice with defects in any component of the extracellular matrix, they tend to have a tendency to develop emphysematous lungs, and those emphysematous lungs can burst and present as pneumothorax. And we see that in about 10% of patients, for example, with Marfan syndrome. And we see it subtly differently in Loeys Dietz and vascular Ehlers-Danlos. Now, I've got at the end this Donald Rumsfeldian term, unknown unknowns. We know, actually, that 75% of people with familial pneumothorax will not easily fall into one of these groups. So we struggle to make a diagnosis in 3/4 of people with familial pneumothorax. And this is where the 100,000 genomes comes in. And this is still very much a work in progress. We were rather late to the 100,000 genomes, the respiratory Gzip. We were only in for about the last 18 months of recruitment. So we've recruited relatively fewer compared to some other conditions, and we had to make our inclusion criteria quite broad. So patients and families simply needed to have a history of a familial pneumothorax, ideally a primary relative, first degree. And we wanted most of these patients at least to have gone through the basic clinical assessment, which I've already told you about, to exclude these obvious causes, to exclude obvious Birt-Hogg-Debye, to exclude obvious Marfans, etc., because we didn't want to be using up valuable sequencing time. The first, I think, very useful thing about the 100,000 Genomes Project taking on familial pneumothorax was to develop the NHS first gene panel for pneumothorax. Up until then, there were no gene panels available for this condition. So using PanelApp, which I'm sure everybody on this call is familiar with, we generated a nice long list of potential genes which have been shown to cause pneumothorax either in humans or in animal models. After appropriate scoring by other members of Genomics England, we're left with these 10 genes, which became our tier 1 genes, including, for example, folliculin, which is the causative gene in Birt-Hogg-Debye syndrome. As with the other gene panels in the 100,000 Genomes Project, as of August 2020, this became an available test as a gene panel via the Genomics Test Directory. So this is already, I think, a deliverable of the 100,000 Genomes Project, is that we've now provided both geneticists and respiratory physicians with ready access to a useful tool. As I said, we were only joined quite late, so we were only able to recruit 33 individuals who fulfilled those recruitment criteria. And you'll see that there's a reasonably even mix between men and women. Usually pneumothorax is seen in men, probably because they're taller and there are mechanical effects to cause pneumothoraces. But when we look at familial pneumothorax, there is a more even split. So the first question we wanted to know is, are we missing known genetic, missing known syndromes? Are these form fruits of Birt-Hogg-Debye or Marfans who are not presenting clinically in the normal way? So looking at these tier genes, tier 1 genes, bearing in mind what we've just heard that we may have missed some, but going by the data that we had available, we were only able to find 1 individual out of 33 who had a pathogenic variant in a tier 1 gene. And this turned out to be a woman with Birt-Hogg-Debye syndrome, a pathogenic mutation in folliculin. So we went back to the case to see, should this person have been diagnosed? She had a mother with pneumothoraces, no family history of renal cancer, no skin changes of Birt-Hogg-Debye syndrome, and only 2 very small cysts. This is one of them. And I know the radiologist who's still beating themselves up about not reporting this as possible Birt-Hogg-Debye, but it's a very small 1 of 2 cysts on a fissure, and it could easily be reported as a small patch of emphysema. So What this immediately tells us is that clinical assessment with patients with familial pneumothorax is very efficient at identifying these known syndromes. It's more than 95% sensitive. And this has relevance for existing clinical services. But it does also mean that the large majority of those 75% of familial pneumothorax patients in whom we can't give them a diagnosis, we are missing something. And this is work that is still ongoing and will actually require further recruitment to other studies. But to lay out the problem, we have, if we look at all of the available pathogenic variants, we have a small number which are seen in many of our patients, or we see many which are unique to individuals. And so is this an unknown syndrome, or are these many ultra rare conditions? And it's more likely to be the latter, or even worse than that, to be polygenic. This is one of the cases in which we think we've identified the problem. One of these twins, who had, they both have mild learning difficulties, cardiac defects, dysmorphic features, and spontaneous pneumothoraces, they turn out to have a mutation of a highly conserved residue in a DNA methyltransferase, and we believe as a result that they have Tatum-Brown-Raman syndrome, which hasn't previously been associated with pneumothorax, and we're currently working with other colleagues to determine if this is a real association. But another deliverable of the familial pneumothorax Gzip is the development of a rare disease collaborative network for familial pneumothorax. As of last year, NHS England has commissioned 2 sites, so Cambridge and Glasgow, to run for the UK a rare disease collaborative network in familial pneumothorax to improve the diagnosis of these known syndromes. We allow access to clinicians from across the UK to an online MDT, which is held regularly, and the referring clinicians simply need to fill in and send us the imaging and the information from this pro forma, and we will discuss these cases, and it's both educational and, we hope, helpful to the patients, so to the clinicians as well. So back up now, as getting towards the end of my introduction and going towards the more important part, which is Fiona's perspective from the patients. We work very closely with patient groups, particularly some rare disease charities, particularly the Myelofibrosis Trust and the Bertrand Hogvay Foundation. When we launched the pneumothorax rare disease collaborative network last summer, in order to make it more readily recognizable, we worked with these charities to do something of a social media blitz. And this is one of the infographics generated by the Bertrand Hogvay Foundation. We decided that the launch day, 30th of June, was going to become World Pneumothorax Day. And although this was done slightly lightheartedly, during the day, many patients and many clinicians around the world came on board, and World Pneumothorax Day became a thing. And this made these rare diseases, they gave them a larger profile. And you can visit the Myelofibrosis Trust website and see some blogs by some patients and some interviews. So I'm going to wrap up my clinical introduction, thank the other people in the respiratory Gzip and the Plural Service in Cambridge, stop sharing my screen and hand over to Fiona.
Thank you, Stefan. Thank you for the big up as well on time. Hopefully I'll be as interesting as everybody thinks I'm going to be. So can everybody hear me? Is that okay? So thank you to Stefan and to Jillian for your kind words. So I'm going to talk to you today a little bit about how patients participated in this research and the benefits that they saw, and give you a few stories from some individual families, patients that participated. And then I should give you a few tips on how you can get patients on board with your research. Now I've just got to work out how to change my screen. Here we go. So how patients help this research. So at the centre of this research, as Stefan said, is the patients. The patients that give up their time, they give you their samples, and they often travel a long way, numerous occasions, to take part in research. But they can also offer other things for you. So they can help you with reviewing research applications, and they can help you actually write lay summaries, which I think I've counted, helped something like 20 plus lay summaries. They can help you prioritize your research proposals, so they can help you maintain the balance between research that can help you now, patients that can help now, but what that can help patients in the future. And obviously Stefan's research was really about diagnosing people, giving them the diagnosis, but patients can help you to work out what kind of research they want you to do, so and keep the balance between what the patient wants and what the clinicians are interested in. So that patients can give you an external perspective to your to research proposals and your meetings. So, for example, in my work life, I'm a project manager, and obviously I can bring those skills to the table when I come to meetings. But really importantly, they can help share their real life experiences with you about how they live with the condition. And sometimes it's quite often the anecdotal things that people tell you about having the condition that might get missed out in a clinical or a research environment. And all of us who work in patient advocacy have great relationships with other patient groups. We're quite a small rare disease community, and we do talk to each other. So, and we can collaborate together, and that can be useful for you in your research. But the other thing that we can do is we can introduce you to other patients, and we can help you recruit and disseminate the results of your research. But how did the patients benefit from this research then? So having a diagnosis for a condition like BHD is really important to the patient, and it makes them understand their prognosis. They can plan for the future. They can think about whether other members of their family have to be screened for the condition. So BHD can affect multiple levels of the family and different generations, but they can also access the appropriate care to recognise the symptoms. So obviously, as Stefan talked about, there's kidney tumours for BHD which need to be managed appropriately, but if you have Marfan syndrome, you can have aneurysms, and if you have regular surveillance scanning, that hopefully can pick those up before they become a problem for the patient. But obviously, once the patient has been diagnosed, they can join research projects, like other research projects other than for the 100,000 genomes. So I think it's really helpful that they do have a diagnosis. But you know, as a parent of 2 children with a rare condition, having access to a peer-to-peer support again is also really, really helpful. So what did these patients learn from the research? And obviously they're really relieved to get a diagnosis, and it's fantastic as a clinician that you're able to diagnose a family through the 100,000 Genomes Project, and it's a big tick for the project, but the diagnosis will probably be the biggest news the family has ever had to deal with in their life, and you need to be really careful on how that information is given to the parents or to the families. So one patient told us that he had suffered PTSD from how he was given the diagnosis, because the doctor was so excited that he'd managed to find out what was wrong with him, and what was discovered, what was causing his issues. But you know, it can be a really huge problem for the patient. So we just need to make sure that who delivers this is done with care and empathy, which sounds very obvious, but it's not always obvious when you get excited as a clinician. And then the other thing is that, you know, you think about the ongoing surveillance of these patients. Patients can get very anxious about having their annual or their twice a year scans, and you need to just be aware that, you know, although for you it's a positive, and it is a positive in looking after these patients, we need to be a bit careful about how we do the surveillance imaging and making sure that their mental health is okay as well. But everybody wants some hope, and by people getting a diagnosis, there is some hope for the future, that the more we learn about the genetics of the disease, the more likely we might be able to find other cures and treatments for it. So I talked to 2 families about their diagnosis, and this family shared their story with me. Mum had her first lung collapse when she was aged 15, and then her daughter had one when she was 17, and another daughter had them when she was 21, and they were just doing normal things in their lives when these happened. but it took nearly 20 years for mum to get the diagnosis. And she said that this research had been life-saving for her, because she discovered that she did have a tumour which was removed in her kidney. But she said, you know, they had all have had multiple surgeries, and the diagnosis had a huge impact on their lives, because they didn't really know what was causing the problems, and they were really scared it might happen again. Every time they had a pneumothorax, they thought it might happen again, and it's very scary when you don't know what's causing these things, and you can see it running through the family. They said that the monitoring of the associated issues was reassuring, but again, it added anxiety to their lives, as they always knew that they would be having these scans on a regular basis. But the mum said to me that her daughters were active, beautiful, and clever. But having a BHT had a huge psychological impact on all of them. So, but the positives are that they're all very well at the moment. They are being monitored, and mum is now also a patient advocate, and she lives in France and is now setting up, you know, trying to set up a support group to help people in France, so that this doesn't happen to other people. So, you know, this research is spreading wider than we think it is. And then the other family, mum and daughter, have BHD. They were diagnosed through the 100,000 Genomes Project. But what happened with the mum was that she actually had kidney cancer, and after being picked up at a blood test, that she had very high levels of iron in her blood. And then during the surgery, she had a pneumothorax for the first time. And since diagnosis, her daughter has also been diagnosed with it, and now they're also looking to screen the next generation. And again, the whole family are really grateful that they know the genes, but it comes with the problems. And Lady said that they both have anxiety about having the condition and about the regular screenings. And she said she's also not very keen on flying in case they get a pneumothorax, unless she lives in Scotland. She doesn't get much sunshine from there. So, but again, this family too are doing very well at the moment, and they're both patient advocates. Mum is the patient rep for the rare cancer Gzip, and they also run a local kidney cancer Facebook support group. So hopefully, you know, they're doing very well. But I think what I would like you to recommend is that you get closer to your patients, and you get close to your patient support groups, rare disease groups, offer to talk to them at conferences, write articles for their newsletters, like Andrew was saying in the headline news. There's an article in there. You know, they're always asking for people to write things for them. So because it builds trust and makes them more receptive to work with you. So try and identify if you can find a super user, someone who's really keen to help research. I was recruited by a clinician at the Brompton Hospital many years ago to help this, and he saw something in me that I thought I could add value, and I think I probably have. But you know, you can invite them to your team meetings, your research meetings, and you can also get them some training as well. There's some great resources on the NIHR website to help patients. So in summary, and I think I've rushed through this a little bit, but I'm conscious of the time and any questions you might have, but I hope that I've demonstrated that having a patient representative a part of your research will have a positive effect on your research, because patients do want to help, and they want to help say thank you for the care that you've given them. We don't have to have any special skills. We just be able to tell you our experiences and give you our opinion. So I would recommend that you just go out and ask them. So I think that's it. Thank you very much to everybody for inviting me to talk today, and to the families that let me share their stories.
Thanks very much, Fiona. That was brilliant.
Okay.
As ever, I think even just one or two slides are phenomenally helpful when we get to thinking about the techniques that you can employ to find your participant community and how then you can engage with them. So thank you very much. People have been quite shy in the chat about this particular session, so we haven't had any Q&A yet, but I've got a question for each of you, which hopefully will help us sort of spark a few of the thoughts. The question I've got, first of all, is for Stefan, really. Can you describe a little bit about the kind of involvement that you and Fiona have had together in delivering the work of your respiratory Gzip, please?
Absolutely, yeah. So I've known Fiona for as long as I've been on the respiratory Gzip, because the respiratory Gzip meets still monthly. Even now, we meet monthly to discuss projects, to discuss how to really leverage the results from the 100,000 Genomes Project, and clinicians from across the UK dial in. But every single month, Fiona is there, and we massively value her input. You know, she is as much a member of the team as any of the geneticists or any of the respiratory physicians. You know, we have a huge amount of respect for her, and she's already alluded to the fact that our lay summaries for all of our grants go through her desk and have been improved vastly as a result.
Thank you. And Fiona, from your perspective, you've shared a number of tips there about things that can help get researchers exposed to the participant communities, but have you got some tips as well about, you know, how you keep that relationship going?
With the doctors that I work with, well, I'm obviously part of the Gzip, but I'm also on lots of research panels. So I sit on lots of panels for particular research, and so I attend lots of meetings, which it used to be face to face, which I have to say was very time critical, and so you can only do a certain number of if you have to go to London for a meeting, for an hour's meeting. But now it's all done on Zoom or online. It means that I can attend more, actually. So yeah, but most of the work that I've been doing over the years is just by meeting people at conferences and things, and people say, oh, actually I need a patient view on this, but I was going to say, as I tried to allude is that all the other people I've met, everybody wants to do it because they want to improve the lives of their families, or they've had a bad experience and they don't want other people to have the same thing. And I think that there is loads of people that would be more than happy to help you in your specialised disease group, and I'm more than happy to give you specific tips of people, you know. So I've helped other people set up support groups around the world, you know, and I think that's really important. But they're your first port of call, I think, if you want to find somebody. But yeah, I mean, I'm not so involved with the PCD support group on a day-to-day basis anymore, but I'm still very active on LinkedIn and things, and so if anybody wants to find me, and you've got my contact details, I'm more than happy to talk to people specifically about what sort of projects I've done. Does that answer the question, Jillian?
Yes, thank you very much. Yes, that's great.
Yeah,
great. Do we have any questions from anyone who's still on the line?
Sam said that there is a raised hand in the chat, and does somebody want to unmute themselves and ask their question?
I think that they've now lowered their hand, so.
Okay.
Fiona's put hers up though.
Yeah, I don't know how I've done that. I think I was just looking at who's put their hands up. Yeah.
Well, I think that would be a worthwhile reminder to everybody out there that working with the participant panel at Genomics England, the Gzip team within Genomics England have been able to secure some funding for patient representatives who are interested in working with the different Gzips. There's a small amount of funding available if you in your Gzips need to be able to support the participation of a representative from the community in the work that you're doing. So get in touch with Zan or the rest of the Gzip team on the standard email if you've got any questions about that, and we can help link you up with someone who might be able to help you, or indeed offer some tips from our perspective as well about how we can meaningfully make relationships there, because I think it's important that the researchers, obviously we've heard a lot today about the expert nature of lived experience and the fact that families who've got these conditions can tell you a lot about it. But I think sometimes it is difficult to know what sort of questions as a researcher you might want to ask that family in order to then move forward. And in many cases, what we've been finding is that essentially just sitting down over a cup of coffee and talking about your respective experiences can actually trigger a lot of useful things. So hopefully that's given you a flavour for the sort of thing that Fiona's been doing, and the inspiration to try and replicate that among your own colleagues.
Yeah, I agree. I think I missed this out. I says we used to have, when we were allowed to meet in face-to-face, we used to have patient days, and we would invite clinicians to come and talk at our patient days. But the clinicians would often say they really enjoyed giving their presentations, but it was the conversations they had afterwards with the patients. You know, the stuff that you don't normally talk about when you're in a clinical environment. So if someone make a comment about going swimming, or they'll, you know, or something, and then it will think the doctor will think of something else. It's like, oh, I've never really thought about that. So, you know, it's just those, as you say, the cups of coffee, you know, or the, you know, just by trying to have a more informal relationship with the patient, not in a clinical environment, you can learn a lot from the patients that you probably wouldn't have noticed before. So.
Yes. Absolutely.
Yeah.
And also the idea that although people may not say they're interested in science per se, as soon as it affects them and their own life directly, you can bet they're going to be interested in finding out about that little niche. So
Yeah.
even if someone like me has no scientific training at all, when something arrives in your life that has a scientific basis, you're blooming well going to want to get on and find out as much as you can about that particular niche. And so researchers and clinicians should never worry about being too technical with people. They need to, you know, they're keen to learn, and they're keen to absorb as much as they can from you about that particular niche, because that's what matters most to the people in their lives.
Yeah.
And it really is important for this less formal environment. The less formal chats are also valuable clinically, not only in terms of researchers. We heard about that patient who told us that the clinician, and I have to say it's not somebody from my team, who was so excited, you know, because you know, we are doing research, we are liking to make those diagnoses, but the way that these very sensitive pieces of information are communicated with patients has to take the patient as the most important person, and that's possibly not always done. And by humanizing both the doctor and the patient, you get more sensitivity. Similarly, even though I, you know, from my cancer patients, I know that there's an awful lot of scan anxiety in terms of, you know, when they're coming up for their follow-up scans. I know that you anticipate it. It's just the same when it comes to scan anxiety for echocardiograms in Marfan's or the renal scans in Berthold Bay. And it's not something that the patients will say, I was really terrified about that scan. They were just delighted to know it was, when they see me in clinic, they're just delighted to know it was negative. This sort of patient interactions is really good to learn about the entire lived experience of these people and how we can improve it.
Absolutely. And similarly, people might think that they've got some bad news to share, but actually that might be good news from the patient perspective, because it rules out a whole lot of other things they might have been more worried about. So never be afraid to share that as well. I think it looks like we haven't got any more questions from the room, so we can move back to, well, first of all, I'd like to thank you both very much for your contributions there. It's great to hear, and great to hear as a double act how things work together with you. So thank you again to all of our speakers today. It's been a fascinating session. Really appreciate that you've had your time and your candor in sharing the things that have been important to you in this. There's a few other things we need to tell you about, just to remind you again, in case you missed it at the beginning. There's the next seminar coming up on the 22nd of February, and that's going to be looking at polygenic risk scores and colorectal cancer. There's a Twitter account which enables you to keep up to speed with all the latest news from the Gzip team. It's at Gzip team, easy enough to remember, and a list of useful resources which are available via the Genomics England website. The research portal in particular has the full list of projects which are ongoing using our data in the 100,000 Genomes Project research environment at the moment, and there are lay summaries attached to all of those. Although from a user perspective, I must say it's quite hard to digest some of those lay summaries, so I think there's more work to do there. But there's a lot of useful information for the research community, certainly. All the previous seminars are recorded on YouTube and are available also via the YouTube channel from Genomics England. And a reminder that the next further non-research seminar events are the Research Summit, which is scheduled for May the 4th. Come and meet people in real life again, actually have coffee with them, and let's hear a lot more about the different research projects that have been going on over the last COVID period. This should be a really exciting day, and currently the Gzip team are looking forward to hearing from you with your abstract submissions. So if you would like to present a poster or do a 15-minute presentation on the day, they would love to hear from you with your proposal. And if you can get those in to events@genomicsengland.co.uk, they would be really pleased to hear from you. The other item on this slide is about the Sight Loss in Your Genome seminar, which is coming up on Thursday of this week. That's going to be Thursday, the 27th of January, if you're watching this on catch up. And that's at 5:30 in the evening. We'll be hearing from two of the senior clinicians and researchers who have been investigating within the sight loss genome. And that is an interactive session, so you'll have a chance to ask questions of them as well as hear about their research. That's also been co-produced in collaboration with some of our participant panel members who themselves have a visual impairment. And we're excited to see that seminar series carrying on. Have we any more today, or are we going to be able to wrap up? I think that is all we have. Yep, I think that's all the news from today. So thank you again for everybody for attending. We're 5 minutes early, amazingly, so you get your extra cup of coffee before moving on to the next thing for the afternoon. And it's been a great pleasure to hear from you, and I'm looking forward to the next seminar already. Thank you all very much.